This is Roger Barnett, the Chairman and CEO of Shaklee Corporation, and welcome to another episode of our Doctor is In series. And this is being aired for the first time at our Shaklee Virtual Global Conference, where we have tens of thousands of people from around the world tuning in. And today I have the great privilege of interviewing Dr.Anne Wyllie, who is an associate researcher at the Yale University School of Public Health. But most importantly, she is at the cutting edge of doing research on testing for COVID using saliva. So Dr. Wyllie, thank you very much for joining me today.
Thanks for having me.
Can you share with us a little bit about the research that you're doing with using saliva at testing? And then later we'll talk about how it's being used with the NBA as well. But tell us the insight and how accurate it is and what led you to use saliva as a testing methodology for COVID.
Yeah, sure. So this actually started back
That's okay.
So this actually started back in March when, back in March when we saw that the pandemic was approaching, a lot of researchers across Yale School of Public Health, Yale School of Medicine, Yale New Haven Hospital just really banded together to form the Impact Biorepository. The idea behind this was to be able to build a bank of samples that we could use for research to really, we knew that we were going to have to sort of study SARS-CoV-2, COVID-19 disease progression later on. And what was happening is that we were receiving these samples in our little research lab, and all of the samples that we were receiving, we were checking them all for SARS-CoV-2 detection. And during this time, it already became quite apparent that, we experienced the firsthand supply chain issues of shortages of the nasopharyngeal swab, which was at that point, the gold standard for COVID diagnostics. We knew that our healthcare workers were incredibly, they were rushed off their feet. They were so busy. And the risk to them for taking these nasopharyngeal swabs, it put them at risk having to get up and close to the patients to do so. And, we're also seeing the testing aversion, the more people got tested, the less they actually wanted to have this nasopharyngeal swab. So as we were receiving all of these samples, one of the sample types that we just happened to be collecting was saliva. And since we were testing them all for SARS-CoV-2 detection, it became quite interesting to us that, we started seeing the data come out, and we started seeing that we were having quite good detection of the virus in the saliva samples. And just knowing the issues that we were facing with the swabs, the trouble getting them, even in terms of research, we were really keenly aware that we did not want to be drawing away from the clinical diagnostic supply chain. So we wanted to make sure that, I think we lost some research opportunities because, we knew that the diagnostics was most important at that time. And, also again, the healthcare workers, if they had to go in and take another swab just for research, for research, that was putting them at additional risk. So as we saw that saliva was starting to look interesting, we decided, this could be worth keeping an eye on. And, it became quite apparent to us that, in the end, it was performing as well as, in some cases, if not better, than the nasopharyngeal sample itself. And so knowing that, and, as the pandemic continued, it, really became aware to us that we were going to have to keep sampling people, and that we're probably going to be facing situations where we would need frequent and repeat testing. And again, like if anyone's had that nasopharyngeal swab, I guess, no one would want to be having that, twice a week, or even once a week kind of thing. So we really saw the potential for saliva to get around, a lot of the supply chain issues, the risk, the testing aversion. So we decided to continue working with that and seeing what we could do to really optimize it as a sample type for SARS-CoV-2 detection.
So if I may for a second, just repeat, it's so fascinating because one of the advantages of doing the research at the Yale School of Public Health is that you have the Yale New Haven Hospital system as well. So there's a pool of people. And so the nasal forensic well, I didn't pronounce that properly, swab, which is what people call the brain tickler, right? The big thing that goes up your nose, is uncomfortable. And what most people may or may not realize is that the supply chain, the agents that are required for that are really in short supply. And so to use saliva could be much more scalable at a much lower cost and it's a much more pleasant experience for people to be tested. Is that accurate?
That's accurate indeed. Yes, it is.
Okay, so then, and then just share with us a little bit, in your perspective, do you believe that saliva is at least equivalent in accuracy from the results of the thing that goes all the way up your nose?
Yeah, so we were processing our saliva samples exactly the same way as the nasopharyngeal swabs. So for this, we're doing the RT-qPCR testing, so the standard sort of PCR testing that's being done to detect the virus RNA that's being done in many of the labs across the country. And through that is when we saw that, saliva was performing just as well as the swabs. But we also knew that this was, it was an expensive process, or how could we make it cheaper? And if we could make that test cheaper, but still as good as the more extensive process, this would really help us to make maybe testing more accessible to more individuals and more communities. So, we realized that we had this really, really good process that was going on to get very sensitive detection in saliva. So we thought, how can we remove some of those costs, but still keep that level of sensitivity? So that's sort of led us down our next sort of research track, and also together with the MBA was our development of Saliva Direct, with the idea that now we can almost take just a very, very simple saliva sample and we test that almost directly into the RT-qPCR. And by taking away that middle RNA extraction step, we've managed to decrease the overall price of the cost by about 80 or 90% as compared to sort of the more extensive methods that are out there. So, that was what, really was sort of behind it, just sort of trying to get this price down so that we could get testing more accessible to more individuals out there.
That's amazing. It really is amazing. Maybe we could just ladder back up for a second and explain why testing is so important in terms of dealing with the COVID pandemic. There's sort of treatment protocols, if you get it. How do you make it less severe and hopefully so it doesn't lead to death? There's a vaccine, which there are many vaccines in practice, but to date, it's the fastest vaccine, I believe, is 4.5 years. And obviously, there's never been the extent of global investment in research and demand, so we'll probably beat that, but that's unknown and unclear, which leads to testing. Explain why testing is so important.
I think it's important for a number of reasons. So the first one, it's really going to help us get this pandemic under control. If we can detect cases early enough, we can isolate individuals before they especially before they become contagious and a risk to other people and continuing to spread the virus. If we can identify those individuals early enough, we can stop the chains of transmission of the virus. It can give us a better idea of just what our risk is out in our local communities, how much virus is actually around, and can help us to keep monitoring that status going forward. So, I think, you mentioned the vaccines, even if the vaccine, even if we see one in about 6 months to a year, testing is still going to be important to have alongside that, because, not everyone is going to be able to have the vaccine. We don't even know if this first round of vaccines is going to work in absolutely everybody. And of course, some people just don't want to have the vaccine either. So, while, while some of our population starts to become protected from that vaccine, we're still going to see maybe still future outbreaks. It'll be good for us to know where this virus is and how we can learn to live with it, and what our risks are if we're going back to reopening to our different, reopening our lives a bit, getting back to workplaces, getting back to colleges, getting back to school. If we can have testing together with that can just make us more aware of what we're doing and the choices that we're making.
Yeah. So let's talk about the NBA for a second, because the NBA took a different approach from all the other sports teams and created an entire bubble. And so, and then they used testing to ensure and verify that the people who were in the bubble and exposed to others were free of the virus. So I have two questions for you. One, and your saliva test was critical to that component to it. One, do you think that emphasis of a bubble is really where we will go? In other words, that if we could get the saliva test down in cost and accessibility with agents that you could create little bubbles that when you go into the office, everybody could be tested and you would know that everybody is there is a safe one, or to certain events? That's question number one. And number two, with the NBA, my understanding is that the testing was so successful in identifying people who had the virus that transmission has effectively stopped. And that has created interesting challenges, good points and bad points for you. So can you comment on both those questions?
Yeah, sure. So indeed, I mean, the NBA has done I can say they've done a really fabulous job. And indeed, they've done such a great job that it is great for their staff, their players, that they've managed to sort of stop this virus in their track. But, it would have been, it's definitely restricted our research a little bit, which, for the good of public health, a couple more sort of positive, negative samples would have been interesting to see. But of course, I don't wish any positive results on anybody, of course. But I guess what the NBA has shown us is what the power of testing can do. And if you can have access to frequent repeat testing, you are able to identify these individuals early on. And that's exactly what they were able to do. And I think, indeed, they created these bubbles in these very controlled environments. I don't think that we need to live to that extent in a bubble. But the idea that, if you have this awareness of who is testing positive around you or not, and you can then have the ability to do contact tracing and follow up on that information that you get. Again, this just shows what you can do when you know where this virus is, and you can catch it early on enough before it suddenly just spreads throughout your population, your group of people, and then it becomes a lot harder to control.
So let me ask the questions that seems overwhelming with what one as a consumer reads about testing. The one thing that's frustrating for a lot of people today is that if they do get a test, it takes a really long time to get the results. And that goes to the capacity of the laboratories and the kinds of machines that can do it. And then there's this test and that test, and there's the antigen test and the RNA test and the blood test and the saliva test and the nasal brain tickler test. So if you were to simplify it for us, tell us, how long would it take to get results if with your Saliva Direct results? And the level of accuracy, in other words, some tests you have to already be sick with it, or some you cannot exhibit symptoms. Like, does it work? Does your test work and identify it, whether you're contagious or not contagious, et cetera?
Yes, indeed. So again, where Saliva Direct is still a PCR-based test. So PCR tests that we have out there are one of the most sensitive tests that are out there at the moment. And so by...
Can you explain what PCR means?
Of course. So PCR is a way, so what happens is that you can take your sample and then you can sort of, we do just a very, very simple chemical treatment on our saliva samples, for example, that helps break open the virus, and that helps expose its genetic material or its RNA. And then what happens with using the PCR is that we are able to take this sort of the sample with that RNA exposed, we can put that into the PCR machine, but we put in little bits of, we can target sequences of that RNA that's in there. And so we're able to add primers and probes, which target these very specific regions of the virus RNA. And if the virus is in there, these primers and probes will find that region, attach to it, and then it will emit a light that the machine can detect. And so if there is, say, for example, a lot of virus in your sample, you'll get a lot of light coming out. You can tell that there's a lot more viral load. If there's only a small amount of virus, you'll get a lot less light. This will come through a lot later, and you can then know that there's a low amount of viral load. So it's really looking for the virus RNA, that's genetic material that's in the sample.
And so do you need a PCR machine to be successful with your results as well?
Yes, so this test, it does need to be run in a lab, and it does need a PCR instrument. But what we've done with so one thing about Saliva Direct is that it's not actually a test. So, I mean, we haven't packaged a kit. We're not sending it out to anyone. We're not manufacturing or distributing anything. In fact, when we got our emergency use authorization from the FDA, they did say it's the most unique one that they've issued to date, and that all that we've actually done is that we've developed a test. And so we've developed the protocol for it, which is like the recipe describing how to do this test. And we're allowed to give that recipe, per se, to other labs to also run this test. But what was key in us doing this is that we wanted to make sure that the labs who are going to run this test also didn't need anything new, or that they didn't have to buy anything new. Nothing new needed to be manufactured or distributed. So what we have done is that we've already tested this method in a variety of different PCR machines, because there are a number out there, just with the idea that hopefully these labs can already utilize what they already have available to them and not have to, buy something specifically for this method.
That's fantastic. And how long between, if you have the machine there and you give them the sample, how long does it take for them to give a result?
So this does depend on the lab. I don't want to put any pressure on the labs out there. We can do about 92 samples in about 3 hours with just one technician doing a lot of very manual work. Of course, labs have different processes in place, especially if they're receiving a lot of samples. You can imagine that they might be just overwhelmed receiving all of those. What we're also hoping with Saliva Direct, though, is that we're going to get a lot more of the local labs up and running in the community. So these are a lot of labs who weren't really doing much SARS-CoV-2 testing before this. And so we're actually, empowering these smaller labs to be able to offer more local testing. So we're adding more tests to the testing market each day. And what's more important is that if you've got a local lab and you can actually just get your samples right there, you're not having to ship them, 12 hours across the country, that should also be able to help us get results back. Maybe in some cases it will be same day, but otherwise within 24 hours. And that's what you really need to be able to sort of act on these positive results.
Well, I think it's so exciting. I think your work has shown with the NBA that you can actually create an environment at small scale, but an environment where COVID has been conquered. And I think it gives us hope of getting a sense of perspective of how we're going to get out of the effect of this pandemic, which is impacting so many people from a health perspective and so many people from a financial perspective as well. So I think your drive to make it easy for people, to make it accessible, to make it affordable, and to make it scalable is really admirable. And I just want to say, on behalf of the 2 million members of our Shaklee family around the world, thank you for this. It's just great to be able to shine a spotlight on what I call heroes at the frontier of medicine to help so many people. And then in my capacity as on the Leadership Council, which is kind of the board of the Yale School of Public Health, couldn't be more proud to have a researcher like yourself with the tremendous work that you're doing. And I know that the entire university is proud of the work. So I just want to thank you for doing what you're doing.
Thank you very much.
So everybody, this wraps up our latest edition of The Doctor is In with Dr. Anne Wyllie, truly a pioneer in groundbreaking testing. It's affordable and accessible and simple and easy for people around COVID-19. And again, Dr. Wyllie, thank you for your time and have a great day.
Thank you. You too.